All right, so I just want to briefly mention the exam from last week. I'm planning right now on Wednesday going through the 4 or 5 most missed questions, so stay tuned for that. I can't do that quite yet. I think for a lot of you said, oh, I haven't taken an in-person exam in a really long time. So I think I maybe should have addressed a little bit harder that I did intend everybody to take it online. I had a number of people that kind of flippantly, oh, I'll just take it at home. And this is an in-person class. So if you work every day at 1:30, then you're going to have to figure out a way to get out of work to come to class. So there are reasons that I might let you take it online, mostly for health concerns. If you're quarantined, you know, for an extended period, then we do have that option. But you know, if you're whatever, something comes up, you know, go into a snowbank or something like that, then come in the next day and we'll do an in-person. So I'd much rather come in and do a makeup a couple days later than just, you know, letting half the class take this online. So trying to just kind of keep the integrity of the exam. Everybody signed up for an in-person class. The other sections are all hybrid, so you had that option before. So, and I think there are advantages to taking it in person, right? You can scratch out the answers on a multiple choice that you know aren't true. So, you know, probably a little too late, but that was my intention. The average was a 74.5. So I ended up kind of Wednesday night, kind of trying to decide whether to curve it a little bit. That's with a 1% curve. So you can see the grade distribution is maybe not necessarily what you would think that it would look like, but that's what it was for the first exam. You can, you know, during office hours, I would encourage all of you, I know that would be a terrible backup, to come look over your exam, see the things that you missed, you know, sometime in the next few weeks. It doesn't have to be this week or today or tomorrow or whatever, but sometime in the next few weeks. Sometimes I can do that over Zoom too. It's a little bit more cumbersome that way, but I have done that already. So again, we'll revisit that a little bit on Wednesday and hit the ones that were most missed. And while I'm thinking about it, tomorrow I have to go get a COVID test during office hours. All of you should be getting tested every week since you're on person, but I try to stick to that, and there was no other time for me to do it except during office hours. So at 9 tomorrow, I have to run over real quick if you were planning on coming in then. Okay. So a week ago, that lecture, remember we started metabolism. We went through all of respiration in that lecture. So we really covered a lot of material kind of in a very quick way. And if you happen to glance at the syllabus lately, we are actually quite a bit ahead of where I thought we might be right now. I'm not worried about that at all because I know I left less time for things coming up. So if we get a little bit ahead now, that's not a problem at all. But we'll take some time here let's do a couple of practice questions. So here's a question about the Entner-Dodorov pathway. So which of these is correct concerning the Entner-Dodorov pathway? And I like to ask questions like this. Which pathway does this? Which pathway does that? So again, we're thinking kind of big picture with some details sprinkled in here. So which is correct? Is this another name for glycolysis? Is this pathway common to most forms of life? Does it produce half the ATP as glycolysis? Is it an anabolic pathway? or is it carried out at the same time as glycolysis? All right, so only one of those is correct. You remember Entner-Doderoff, that is an alternative pathway to glycolysis. So that other name we had for glycolysis was Embden-Meyerhoff, not Entner-Doderoff. So that one's not correct. This is a pathway only used by bacteria. So not common to most forms of life. And it's only some bacteria. Probably not very many of them do this instead of glycolysis because they're missing one of those glycolytic enzymes. This is not an anabolic pathway. It is, we're breaking down glucose into smaller molecules. Remember, it's a catabolic. So the difference between anabolic and catabolic, we're getting ATP out of it. It's carried out at the same time as glycolysis. The bacteria that do this are either going to do one or the other. They're not going to do them at the same time. We talked about how there may be some organisms that would do this pathway when they're grown on a certain media instead of glycolysis, but they're either going to do one or the other. So correct answer is C. Half the ATP is glycolysis. All right. One more question. So we went, like I said, we went through all of respiration. We haven't done fermentation yet, and that's one of the possible answers. So you can be sure that that's not the right answer. So this pathway generates electrons that drive biosynthetic reactions. Right. So the correct answer, there's actually two correct answers. Entner-Doderoff actually does that as well, but that wasn't one of the possible answers. This is the pentose phosphate. We do pentose phosphate in some of our cells. It's another alternative pathway to glycolysis when an organism's needs are more precursors for anabolic, for biosynthetic pathways. So an organism like a bacteria might be doing pentose phosphate at the same time as it's doing glycolysis, for instance, or at the same time as Entner-Doderoff. Not going to be doing all three of them at once. All right, so that's just an example of some of the kind of questions that I would ask about this chapter. But we need to finish up metabolism. We need to talk about a kind of respiration that is unique to bacteria. We do not do anaerobic respiration, obviously. Basically, so it's going through the respiratory pathway, the glycolysis, Krebs cycle, electron transport chain. The difference is there's some electron acceptor other than oxygen that's accepting those electrons. And usually it's going to be an inorganic ion. So remember, something's organic if it has carbon-hydrogen bonds. So inorganic doesn't have carbon-hydrogen bonds. Okay, so some examples of ions that can act as final electron acceptors. And remember our diagram here. I don't think I posted this again, but we're talking about here at the end of the electron transport chain, something other than oxygen is acting to pick up those electrons. Okay, and it could be some examples of ions that could be nitrate is a possible electron acceptor. So if you are in lab, last week you inoculated a nitrate reductase test. Okay, that enzyme nitrate reductase reduces nitrate to nitrite. I'll just write that here. Okay, now obviously that's not a balanced equation. There's actually, let's see, let me just erase this for a second. I'll write in the whole equation here. You can, to balance it, or we wrote in the whole thing, we'd write in those electrons and protons that enzyme nitrate reductase, again, you know, if you're in lab, just making a connection there. And you end up with nitrite and water. Okay, so That's a reduction because actually the oxygen here from the nitrate, it's losing an oxygen. So that's another way. Loss of oxygen is also a reduction. Okay, and just to kind of, let me just skip ahead here a second. I didn't order these quite right. I didn't post this slide because I didn't want it to be something that you really worried about, but that enzyme that if you were in lab, you tested for, nitrate reductase, that's an enzyme that's right there in the cytoplasmic membrane, right next to those electron carriers like the cytochromes, this iron complex here. It's an enzyme that's right there that allows nitrate to act as a final electron acceptor. All right, so if an organism has that enzyme, then allows them to respire anaerobically. Obviously, an enzyme that we don't have. Okay, some other examples of electron acceptors that can be used. Iron is one. Sulfates. And in this case, hydrogen sulfide is going to be the end product. If you've ever been to Yellowstone Park, that kind of rotten egg smell that you get, that's hydrogen sulfide. So that's bacteria making that really distinct odor. And this is something that we test for later on in the semester in lab. It's not produced in large enough quantities for us to smell it. But again, that's an end product of anaerobic respiration. Carbonates is another one. Some organisms reduce carbonate and end up with CH4, methane. Okay, very potent greenhouse gas. Methanogens are organisms that are able to produce methane. And here's an example, again, another slide that I didn't post. This is just a picture from your book, but this is a mechanism to collect methane from a landfill. So this is a reason that, in landfills, they're constantly, covering up the garbage, covering it up with dirt, making these huge piles. Okay, so there's anaerobic organisms in there that are decomposing that trash. That's why you see vents coming up through a landfill to try to vent off that gas that results as a result of anaerobic respiration. And some landfills will have a contraption here to catch that methane gas and then utilize it as a renewable source of energy to power whatever electricity they need at the landfill. Often, and if you go to the landfill just south of Grand Rapids, they do this on a very small scale. It's not really financially productive at this point. In other words, the investment that you have to make for all this machinery to trap the methane and convert it into electricity isn't really going to make you come out on top financially. But there's potential for this in the future. Most of the time, they just burn off the methane, so it doesn't contribute to, the global warming. But okay, so really the other point, there's one other point that I want to make about anaerobic respiration besides, hey, this is bacteria do this, we don't. One other point that I want to make, and then I'm going to try to explain why that is. So I always think we remember things better if you kind of understand why that happens rather than if I just tell you. But the point I want to make is that organisms that respire, or let's say that respire anaerobically, and remember that means they use something other than oxygen as a final electron acceptor, always gain less ATP than if oxygen is used as a final electron acceptor. Okay, so the pathways of anaerobic respiration, like I said, is glycolysis, Krebs cycle, electronic transfer chain. The Krebs cycle is a little bit altered in anaerobic respiration. It's similar, and they get quite, organisms can gain quite a bit of ATP, but never as much as if they use oxygen. And the reason is due to this redox tower. So molecules, we can actually measure how badly molecules want to give up their electrons. Okay, and we can actually stick electrodes in cells and measure that. Okay, so the more that a substance wants to give up its electrons, we say that's the more negative this measure of voltages. It's called E naught prime. So that E, that little zero was pronounced not in the prime. So the more negative the E naught prime is, the more it wants to give up electrons. The more positive the E naught prime is, the more a molecule wants to gain electrons. So you'll see here, and we can use this to calculate how much ATP would be gained during respiration, depending on what's used as the electron donor and the electron acceptor. And I'll let your biochem professor teach you how to calculate that. But basically, it's the distance between the donor and the acceptor. If that's a great big difference, then you're going to gain lots of ATP. If the donor and the acceptor, there's not very much of a potential difference there, then there's not going to be a lot of ATP gained. So for instance, if here's the NAD, when the NADH is giving up one of its electrons, the distance here to say sulfate, well, that's just a short difference. So if sulfate is used then as an electron acceptor, get a little bit of energy there. And incidentally, this is how we know the order of the cytochromes, the electron carriers, different electron carriers in the electron transport chain, because we can measure, what would be giving an electron to the other one. How about the distance here to the nitrate? That's a pretty long distance. If nitrate is used as a final electron acceptor, sometimes organisms have other enzymes that allow them to further reduce that nitrate. That's why this is a little bit further away. Here's iron, but none of those potential anaerobic electron acceptors are as great a distance here as oxygen. So that's why you always get more ATP from aerobic respiration than what you do from anaerobic respiration, in a nutshell. Okay, so what kind of organisms will respire anaerobically? Thinking about our oxygen requirements that we talked about just a little bit ago. Facultative anaerobes. So remember, those organisms can respire aerobically and also do either anaerobic respiration and fermentation. Aerotolerant anaerobes. Remember, those are organisms that can survive in the presence of oxygen. They have enzymes to detoxify it, but they don't use it. So they're going to get their energy either from anaerobic respiration or fermentation, or both. And then strict anaerobes, right, don't respire aerobically at all either, don't have enzymes to detoxify those toxic forms of oxygen, and they're only going to do anaerobic respiration or fermentation. So what's the difference between anaerobic respiration and fermentation? Okay, it comes down to the fate of pyruvate. Turn on my microphone though. Okay. So let me write that in. All right, so if we go back to our kind of overall diagram here, we're looking at what happens to this pyruvate or pyruvic acid when it comes through, say it's coming through glycolysis. All right, if there is a suitable electron acceptor, it will go through a respiration pathway. If not, then the cell has no choice but to enter into some kind of fermentative pathway. Okay, and in fermentation, the final electron acceptor is typically going to be an organic molecule. So it's something that's typically inside of the cell already. Pretty often it's that pyruvic acid. Okay, so again, if there's no other electron acceptor down here, the cell has to have some way to regenerate this NADH back to NAD. Because if it can't get rid of those electrons somewhere, then even glycolysis will stop. Okay, those oxidation reduction reactions have to balance out. And if you don't get NAD being reduced to NADH, then this oxidation is not going to take place. So those electrons have to get dumped off somewhere. If it's not an electron transport chain, then they're going to dump it off on something else in the cell. And typically, you're not going to get any more energy that way, but at least you get the energy out of glycolysis. Okay, so glycolysis can really be considered part of a respiration pathway as well as a fermentative pathway. So let me write this in here. Fermentative pathways. Oxidize NADH back to NAD to keep glycolysis going. And I'll just write that in here. Usually no additional ATP gained in a fermentative pathway. So get the ATP from glycolysis, but and that's not always true. Sometimes there's a little bit of ATP, but usually not any additional. Certainly not like what we get in respiration. Okay, so your book just gives a couple examples of some fermentative pathways. So I think this diagram is a little bit misleading because typically an organism is going to use one fermentation pathway or another. So usually always going to use one, because that's one way that we can use to identify organisms is to try to understand which pathway they use by testing for what the end product is. So in this case, this looks like the organism is either going this way or this way, and it's only going to do like one pathway or the other. So this simplest, so just ignoring the right side here, this is the simplest fermentation pathway that we find in bacteria, where that pyruvic acid here, it's just accepting that electron. Remember, this is actually 2 electrons and a proton. It's accepting those electrons, and here we see them right here. Pyruvic acid is not getting changed very much, but now it changes into lactic acid. Okay, there's no extra gas given off. Many fermentative pathways have some kind of gas as a byproduct. Not so in a lactic acid fermentation. Incidentally, this is the kind of fermentation that we can do for a short period of time in our muscle cells. You're running that 400, that last 100, it gets harder and harder to move. It's because the lactic acid is building up. Okay, so very acidic. Most fermentative products in bacteria, anyway, they're going to have acidic end products. Almost always. Here's another fermentative pathway, though, that this is yeast carry this out. They carry out alcoholic fermentation. So you can see there's a little bit of gas given off. Usually it's carbon dioxide. It could be another gas like hydrogen. And we end up with ethanol, okay, alcohol. So again, I don't know, there may be some bacteria that do that, but I think that's primarily yeast that do that. Okay. All right. So like I said, there's this lactic acid fermentation. no ATP given off there, but they do keep glycolysis going. Okay, and here's another slide that I didn't post because I didn't want to make you too nervous. Here it comes. But this, I just want to give you an idea that in the bacterial world, there are many different fermentative pathways. There's a lot of variation. And we are glad about that because often I spend a lot of time talking about how bacteria hurt us, but we take advantage of these various end products of bacterial fermentations. Here's the lactic acid one that I talked about. This one that ends in propionate. That's a very distinctive flavor that you find in Swiss cheese. All right, so carbon dioxide is given off. That's the holes in Swiss cheese. In the lab, we'll spend some time later on with a test that determines whether an organism does this 2,3-butaneediol fermentation, or this is what we call mixed acid fermentation. Some of our organisms do one, some do the other, and that helps us identify them when we test for the end products there. Here, this pathway over here, the propionate, actually does give a little bit of ATP. So that's not a hard and fast rule. No additional ATP, no ATP given with the 2,3-butaneediol or the mixed acid. Another one I'll mention, the Clostridium. So we've mentioned that genus Clostridium is one of our organisms that is a spore former. A very interesting genera of organisms, has a wide range of disease. One of them is Clostridium Difficile. So C. diff is a really nasty diarrhea, prevalent people that are in the hospital or nursing homes, people that are on antibiotics that can knock out kind of their normal variation of organisms in their large intestine, and the C. diff that you might usually have there in very small amounts takes over, and again, cause a really nasty diarrhea. This intermediate of the Clostridial fermentation butyrate is supposed to be like the smelliest, beyond awful kind of smell. And some nurses say that they can smell whether a patient has C. diff or not by smelling their diarrhea. So they call it the whiff of the diff, all right? So, kind of interesting stuff there. The whole deodorant industry is based on bacterial fermentation, right? They're growing on our skin in warm, moist places. And if you are in lab, today you inoculated, or this week you'll inoculate some fermentation tubes. So basically these are tubes, they were purple broths when we inoculated them. There's a pH indicator in there. There's different sugars in each one of these tubes. And if the organism is able to ferment that sugar, we look for an acidic end product as evidence that the organism is able to metabolize or ferment. Now there might be some aerobic respiration going on there too, where the end products would be maybe very slightly acidic, but we know there's fermentation going on if they're, yellow like this to really tip that pH indicator to change in color. All right, so we want to see if the organism has the enzymes to say split that disaccharide sucrose into its monosaccharides so that the organism can metabolize it. And if you look closely, there's a little inverted test tube here called a Durham tube. I kind of covered it with my marker, but there's a little gas bubble at the top of that one there, just an indication, kind of a hint as to what pathway the organism is using. Is it using one that produces a gas as a byproduct? All right, so again, just trying to draw a connection if you are in lab. And this is another figure from your book. Don't memorize this. I'm just trying to bring home the point that we benefit from bacterial fermentation. So food products like Swiss cheese, here's some organisms that do lactic acid fermentation, cheese, yogurt, even soy sauce is a product of bacterial fermentation. Of course, yeast ferments sugars into alcohol. Clostridium give us things like acetone and isopropanol, things that, well, you don't think about them as being organic. Acetic acid, vinegar, again, another end product of bacterial fermentations. Okay. So that's pretty much all I got for you there. I just want to bring your attention to a table. Sometimes these tables are beneficial to kind of quiz yourself. So, cover up some of the words and see if you can fill in, this is kind of like, okay, are you getting the big picture type of thing? So we talked about all these things and you could cover it up and kind of quiz yourself, okay? And I put that in there twice by accident. Okay, so again, I refer you to the learning objectives to kind of pay attention to. We're not getting super detail oriented for that metabolism chapter. Try to think more kind of big picture. And hopefully the homework questions will be helpful with that as well. All right, so moving on to chapter 7, we are going to spend at least a few lectures in chapter 7 and microbial genetics. The homework for this is a pretty long chapter, so the homework assignment reflects that. There's a lot of animations in the homework. Don't just skip those and try to answer the questions without them. They're there to help you visualize things. So take advantage of that. Don't be in a hurry when you go through those homework questions. The more times you see things, the more likely you are to remember it. So we're going to start with the structure of DNA. Hopefully a lot of this is review. You've had it from other classes. We are going to be focusing primarily on, obviously, bacterial genetics. So, right, there's a lot of sameness between bacteria and eukaryotic cells. From time to time, I'll point out how it's different in eukaryotic cells, but we are mostly concerned with bacteria. All right. And then we're going to get into kind of the nitty gritty of how DNA copies itself. We call that process replication. And then how we make RNA from DNA, transcription, and how we translate that RNA into proteins. It's called translation. We also want to think about how the cell keeps from wasting energy. So obviously that's energetically fairly expensive to produce proteins. The cell doesn't want to produce ones that it's not going to use. And what happens when something goes wrong? What is mutation? What are some different kinds of mutation? How can the cell fix it? And some kind of 3 ways of horizontal gene transfer, another way that the cell's genome can end up being altered. All right, so structure of DNA. Sometimes I have students, before we even start, can you draw the structure of DNA? Hopefully you can do that, but hopefully you could draw something like this, right? The backbone of DNA consists of phosphate, sugar, backbone, alternating phosphate. Sugar is a pentose, a 5-carbon sugar, deoxyribose. And then the bases are bonded to those sugars, right? So there's 4 bases, 2 purines, 2 pyrimidines. The purines are adenine, adenine, and guanine. Okay, they actually, it's not drawn in this much detail here, but they actually have 2 rings. And then the pyrimidines are cytosine and thymine. Okay, so when an adenine is bonded to a thymine, there's always 2 hydrogen bonds. And when a guanine is bonded to a cytosine, there's always 3 hydrogen bonds. So we're going to pay attention to that in spots of the DNA where, say, replication begins, spots where those bonds need to be broken between those bases. There will often be a spot where there's lots of A's and T's because it's a little bit easier for the cell to break 2 hydrogen bonds rather than 3 hydrogen bonds. Okay, and then this is your book's graphic for thinking about structures of DNA. Obviously, it's not something that is going to serve our purposes to really memorize that. But you should kind of have some idea, the structure of DNA, double helix, right? Watson and Crick elucidated that in a paper in 1953, I believe. Probably a lot of other people contributed to that, but they didn't give credit to Rosalind Franklin, for example. They didn't do any experiments themselves. They just used experiments that other people had done and then kind of thought about it in a new way and wrote this one-page paper that's probably one of the most famous papers. It has a major groove and a minor groove that can make differences to what sorts of things bind in certain places. And DNA, of course, is anti-parallel. That means that one end has the 3' end matches up with the 5' end of the other strand, of the opposite strand. So what is that 3' and 5'? What does that mean? Well, we number the carbons on that deoxyribose sugar. So if you look here, we always number, let me just change colors here. We always number that first carbon, the one that's bonding to the base. That's the 1' carbon. And then moving in a clockwise direction from the oxygen, we get the 2', 3', 4', and then the 5' carbon is the one that's not in the ring. It's actually sticking out from the ring. So that's why this is the 5' end with the 5' carbon, and this is the 3' end with the 3'. Okay, so the other strand is the opposite direction. Here's the 1' that's bonded to the base. And then again, moving clockwise here, 2, 3, 4, 5'. So 5' and 3'. Okay. The other thing that the notation that's a little bit cumbersome, or the nomenclature, I should say, the difference between a nucleoside and a nucleotide. A nucleoside is the base and the sugar, and the nucleotide is the base, the sugar, and the phosphate. So yeah, they're close to the same thing, but there's a little bit different in the difference in the terminology there. Okay. All right, so we talked a little bit about the structure of genomes a little bit when we were thinking about structures of bacterial cells. For prokaryotes, as you remember, we said that there's 1, sometimes 2 chromosomes. Well, there's usually going to be 1, and it's usually circular. Well, there are all sorts of exceptions here, but when you have a circular chromosome, it does protect. There's no ends to protect, all right? So it's a little bit more stable. Again, sometimes there's a couple chromosomes. They're always haploid. All right, so there's 1 copy. And they're found in the nucleoid region of the cell. Okay, so in this picture, there's not a membrane around it, but it's all kind of, centered or, well, in one spot in the cytoplasm here. And it's kind of hard to see, remember a transmission electron micrograph is a cross section, so it's kind of hard to see that this is one big circular chromosome there for sure. All right, compare that with eukaryotes. There's usually going to be multiple chromosomes. They're going to be linear usually and diploid. Okay, so for instance, we have 23 pairs, 2 copies of each chromosome. They're going to be found in the nucleus. And keep in mind, though, the DNA of mitochondria and chloroplasts is circular. So further evidence of that endosymbiotic theory. All right, in bacterial cells, they not only have a chromosome, but oftentimes will have multiple plasmids. So we mentioned the structure before, small circular molecules of DNA. They typically will not replicate on the same schedule as the chromosomal DNA. And they typically will encode for traits that are not absolutely necessary for the survival of the cell. So you're not going to find those glycolytic enzymes on plasmid DNA. Those are going to be on the chromosome. What sort of instructions might the plasmids carry? Here's some examples of different kinds of plasmids. You could have a fertility plasmid, and we call that an F plasmid. Okay, we're going to talk about those kind of at the end of the chapter. That enables the organism to do this horizontal gene transfer process called conjugation. They have a pilus to facilitate that if they have an F plasmid. Resistance or R plasmids carries genes for resistance to some kinds of antibiotic. So how do they make the cell resistant to the antibiotic? Just by carrying some information. We're going to talk more about that in the coming weeks, but it might encode for some enzyme that makes the antibiotic not useful anymore. Maybe just breaking some bonds, something like that. Bacteriocin plasmids carry the information for a cell to make bacteriocins. And we usually consider those kind of a proteinaceous toxin. Some kind of a protein that allows the cell to kill other very similar cells. Scientists disagree. Sometimes some scientists argue that they're more of a really narrow spectrum antibiotic, meaning narrow spectrum, meaning it only kills a very certain kind of cell. And again, you can see how that would give the bacterial cell a better chance at survival if it could kind of outcompete wherever the medium it's in. And then one more example of virulence plasmid. That would be a plasmid that encodes some sort of protein that would give the cell a better chance to establish itself in a host and cause an infection. So it might be something that makes it outwit our host immune system. It might be the information to make some kind of a toxin, something that would give it a better chance to establish an infection. All right. So let's talk a little bit now about DNA replication. So Watson and Crick didn't really obviously do any experiments. They weren't sure how DNA was replicated. They had a theory or a hypothesis how they thought that it might happen. And at the time in the 50s, so it was the early 1950s, there was really three theories as to how DNA got copied. But they didn't know which was true. It could be semi-conservative. So if you've printed your notes black and white, this is going to be a little bit harder to figure out what's going on in this experiment. But with semi-conservative replication, that means that one strand of the parent DNA is conserved in the daughter. So you can see here, if the parent is in red, the daughter, the red strand was from the parent, and then the pink strand is the new one that was made. So each daughter strand has part of the parent. So that's semi-conservative. They thought, well, it might be a conservative replication, meaning that the parent strand is conserved, it's the same. Somehow that unwinds, and there's two the two daughter strands then wind together. So you have, a brand new one and an old one. Or there could be some kind of dispersive replication where the parent strand is kind of chopped up in pieces, and you have, bits of old and new together into the, whatever DNA results from that. Okay. In your book, it doesn't, I don't think, mention this experiment at all, but it's been called the most beautiful experiment in biology. So I thought we'd address it. I think it's good to look at experiments that are famous from time to time to see the design of the experiment. And this experiment, done in 1958, was called the most beautiful experiment in biology because not only did it prove which of those three hypotheses was correct concerning DNA replication, but it also disproved the other two. So not only is this right, but these two can't be right. Okay, so this method of replication was very readily accepted, just from one very nicely designed experiment. So I posted a couple links to animations that kind of walk you through this experiment, but here's the crux of it. So they grew some E. coli in a heavy isotope of nitrogen. Okay, there's lots of nitrogen in DNA, nitrogenous bases, okay, lots of nitrogen. So they grew it in this, it's not radioactive, just a little heavier nitrogen. And then when they isolated the DNA and spun it down in a centrifuge, they got, a band of a certain, at a certain place, in some medium that kind of keeps it dispersed. And then they switched the E. coli and grew it on the regular form of nitrogen. Okay, so they found after they spun those cells down after one generation, so they had to know the generation time, of E. coli under certain conditions. And after one generation, extracted the DNA, spun it down again, and they found a band a little bit higher than that original heavy one. Okay, and then they went out a second generation and found that same hybrid band, and then another band that was a little bit lighter. Okay, and as they went out more and more generations, this lighter band got heavier and heavier, and this hybrid got a little bit worse, or a little bit lighter. All right, so what could they, from that, they could conclude that this is the way that the DNA has to be replicated. It has to be semi-conservative, a mix of the old and the new. And I went to a different source that kind of maybe makes that a little bit more clear. So with the semi-conservative here, you get just one band after, this hybrid band, and then you go another generation and you get a hybrid band and a lighter band. So we know that's what we found. So we know that that's what it is. We could discount the conservative method, because if it was conservative, after one generation, you would have two bands. This would be the heavy one on the left and the light one on the right. You wouldn't have a hybrid. So we knew already that method couldn't be correct. If it was dispersive, right, there would still be a hybrid like that semi-conservative. So you had to go out another generation, and this would still just be a hybrid. You wouldn't start to get these two lighter bands here. All right, so that's how we knew that one wasn't correct. So again, I posted two different animations. The first one is a little bit more concise, and I like that one. But when I went to go back and check that link, I couldn't play it on my computer because my Adobe Flash was out of date or whatever. So if you can't get that one, here's one from YouTube that's nicely explains it as well. All right, so if we start to think about what's going on at a more molecular level. Okay, DNA replication is just essentially a polymerization reaction. You have lots of nucleotides that are being joined together to form one big long lots of monomers forming one big long polymer. And it's kind of interesting. You think about, oh, it's these nucleotides, but actually the nucleotides carry with them their own energy for polymerization. They actually don't just carry one phosphate, they actually carry 3 phosphates. And again, the nomenclature is a little bit cumbersome here. If this was a guanine base, we would call that a guanosine triphosphate deoxyribonucleotide. So when it's one of these monomers that's floating around in the medium, we put this little D in front of it, and that means it's a, it's got these 2 high energy bonds that actually break to power that polymerization reaction. All right, so here's your book's kind of simple, how this works. You've got an existing DNA strand here. Of course, this is just one strand. You've got a template strand over here, so it knows what base to bring in. That triphosphate nucleotide binds to that 3' hydroxyl group. We're going to pay attention to the directionality because that's always been a confusing thing. It needs that 3' hydroxyl group for those triphosphate nucleotides to bond to. And again, this is energetically very favorable because it's cleaving off not just 1, but 2 phosphates to make that a very fast, favorable reaction. Okay, and now we get a longer DNA strand. All right, so if we look at, before we get to like the enzymes involved, okay, in bacteria, the site of the beginning of replication, we have a special name for that. Okay, so the site for the start of replication in the chromosome is called the origin. Okay, so that's a term to remember. So in this figure, figure 7.7, this is not my favorite figure because the 2 colors they use are almost the same color. So they start out with, this one's both strands are blue. And then the daughter cells, one's purple and one's blue to kind of, okay, this is semi-conservative. Okay, but the site where DNA replication begins, that's called the origin. So they've marked that with the little spot there on the chromosome. In bacteria, there's only going to be one origin. With eukaryotic cells, they'll have multiple origins because there's so much more DNA in eukaryotic cells. If you've ever seen, a diagram that looks like this, okay, multiple places, again, in eukaryotes where replication forks open up and replication begins. Okay, so in bacteria, there's only going to be one. Eukaryotes, multiple origins. There's too much DNA to not have multiple starting sites there. Okay, and it proceeds in both directions. So you can kind of see that here from the diagram. It's going off in both directions from where it opens up. How are those DNA strands pulled apart? That's a job of a particular enzyme called helicase. Okay, it's going to break the hydrogen bonds between complementary bases. So the bases on one strand that are bonded to the bases on the other. Okay. And when it proceeds in both directions, sometimes this here is called a theta structure because this looks like a Greek theta. So sometimes you hear that. Again, both directions, and then you end up with 2 chromosomes. Okay? And Eventually the cell will divide, one chromosome goes with one cell, one chromosome with the other. All right, so if we do a close-up of a replication fork, so if we kind of zoom in on this spot of the chromosome dividing, we would come to figure 7.6 in your book here, and think about some of the other enzymes that are involved here. All right, so here's that replication fork. We've got the DNA helicase. That's this kind of greenish tube that they show here. That's the enzyme that pulls the two strands apart, and some stabilizing proteins so those strands stay apart. Okay, this DNA up here that hasn't been unwound yet, we would say that's upstream of the replication fork. And in eukaryotes and actually in archaea as well, that DNA is wound around histone proteins. Okay, so that's not the case in prokaryotes. They don't have histones, but again, just pointing out a difference there. Okay, so the replication fork is held open, and then really the enzyme that's the real star of the show here is the DNA polymerase 3. Okay, so there's multiple copies of this. DNA polymerase 3, there's actually at least 5 different DNA polymerases. They were labeled, they were numbered in the order that we discovered them. So that's kind of unusual that the third one is the most important one. But what that enzyme does is adds those triphosphate nucleotides to the 3' end of the growing strand. Okay, so just thinking about the directionality, you know that this strand, the DNA polymerase, is moving in this direction because they're anti-parallel. So the new strand, this would be the 5' end, if given that this is the 3' end, and this is going to be the 3' end. Okay, and that DNA polymerase is always adding on to that 3'. There's a hydroxyl group there that it always adds the new deoxyribonucleotides on. Okay, the other strand actually has to go adding on to the 3' end as well. So it has to go in pieces here. All right, which is a lot more complicated. Okay, let's see, what else about this enzyme? It's the speed is actually incredible. 500 to 1,000 nucleotides or triphosphate nucleotides per second. Astonishing speed that happens there, especially when you consider that it proofreads as well. So it can kind of check to make sure the enzyme that it put the correct base in, the one that's complementary to the other base. So it's extremely fast, and it doesn't make very many errors, which is exactly what we need, right? If the instructions for something are wrong, every time you make something from those instructions, whatever you make is going to be wrong too. So we need that fidelity to be really high when DNA is replicated. Okay. All right. So what else? Actually, that DNA polymerase 3 can't just latch on to the strand and start polymerizing. We would say it can't start de novo, so it can't start from nothing. It actually needs a primer. It needs a 3' hydroxyl group to add the new nucleotides on. All right, so we have an enzyme Primase that synthesizes an RNA primer. So that, again, RNA polymerase has something to start from. And this is just temporary. It's maybe like 10 to 20 base pairs, not quite as short as we make it look in this picture here, but pretty short. This is RNA, so you can't leave a little strip of RNA in the DNA. That's going to be excised out later and DNA added. Okay. Okay. Okay, make a note of that because it's made of RNA. Okay, so there's only one primer necessary for the leading strand. The lagging strand, the one that has to go in the other direction, actually has to have a series of primers. And the next picture shows that a little bit better. Okay. So again, synthesis of the lagging strand, Primase makes a series of primers. Okay, so they're in red. The DNA polymerase 3 then synthesizes DNA. Synthesizes the DNA in fragments. And those fragments have a special name. They're called Okazaki fragments. And they're somewhere on the neighborhood of 1,000 to 2,000 base pairs. So short in the scheme of things, but still, that seems like a lot. Okay, and then a couple other enzymes that are important. The DNA polymerase 1 replaces the primer and fills it in with DNA. Again, let's just remember just a real short piece. And then one other enzyme is necessary, DNA ligase, we say fills in the gaps of what that DNA polymerase 1. So the DNA polymerase 1 gets rid of the RNA, fills in some DNA, but the ligase is necessary to add the phosphate groups. So it's a little bit of, kind of glue to hold it all together. You could think of it that way. All right, so there's a number of enzymes to keep track of here. We've got the DNA polymerase 3, the helicase that works up here, the primase, polymerase 1, and the ligase. So this YouTube video here is just a couple minutes long. It's a nice animation that kind of shows the action of all those enzymes and kind of gives you a little bit of an appreciation of how that might actually look. I think there's some animations associated with your homework as well. So, okay. Just a few other, as we're finishing up replication here, a few other things that I want to mention. One problem that the cell has to have a mechanism to deal with is supercoiling. So you can kind of imagine, here's a replication fork here opening up. If you're just opening, and opening, what's going to happen upstream here? That's going to get more and more twisted up. Okay, so just to illustrate that, if you were holding two ends of, some rubber bands that were coiled together, and you kept opening one end, you're going to get what we call this supercoiling. So it's getting even more twisted up. Eventually, you're not going to be able to keep opening this up anymore because the mechanical stress is too much. Everybody's done that. They've sat on a swing when they were a kid and spun around. Eventually, right, you get lower and lower. You can't pull those chains apart anymore when you're twisting up. So the cell has to have a way to relax those supercoils. Otherwise, replication is going to stop. Okay, so it uses an enzyme called DNA gyrase. And we have similar enzymes in eukaryotes as well. The bacterial enzyme is different enough that we actually have antibiotics that target DNA gyrase and don't have much effect on our similar enzymes that have a similar job. But what that enzyme does, we could spend like a whole class picking apart the mechanism of what it does, but it basically makes cuts in both strands of DNA, relaxes, unwinds it a little bit, and seals those both strands of DNA up again. So it's pretty amazing that one enzyme can kind of do all that. All right, so again, that operates upstream of the replication fork. And one other thing I would mention, because this will kind of come into play a little bit later on, there's a good deal of methylation that takes place once replication has finished. Sorry, methylation, did I say that right? Methylation just means you're adding on a methyl group, so that's CH3 side group. And typically in bacteria, it's the adenine bases that get methylated. And that's different for us. I think it's cytosines that get methylated. So that's one difference between, bacterial DNA and our DNA. We think that there's a lot of methylation around the origin. So that's how the enzymes know kind of where to begin replicating that DNA. We're going to see later on how that can be involved in DNA repair, how the cell knows, like, if there's an error, which strand the error is in, and it may play a role in regulation. So DNA that, say, for instance, is highly methylated is more likely to get transcribed and translated, or maybe less likely. So that just might play a role there. Okay, what am I doing? All right. All right, so before we get into kind of the nitty-gritty of transcription, which is the next step as we follow the flow of information, let's remember the central dogma of genetics. So how is information passed in a cell? All right, so we talked about when DNA is copied, that's called replication. That doesn't necessarily have to take place before the DNA is transcribed. Sometimes the way we present it, it makes you think, oh, replication and then transcription and then translation. Well, not necessarily. But of course, the sequence of bases in DNA, that's an organism's genotype. And then when one or the other strands is made into RNA, we call that transcription. Notice that we never go from messenger RNA to DNA. All right, so for instance, if you get a vaccine that's a messenger RNA vaccine, there's absolutely no way that that can become DNA in your cells and that it can somehow change your DNA. Humans don't have any enzymes that makes that possible. That happens sometimes with viruses, but it's impossible for that to happen again with a messenger RNA vaccine. Messenger RNA is translated, and by the way, there's other kinds of RNA that can get transcribed. It doesn't have to be messenger RNA, but messenger RNA is the kind of RNA that is read at the ribosomes and translated into some polypeptide or protein. Okay, and of course, what proteins an organism has kind of determines its traits or its phenotypes. Right, so we're going to follow that information trail. So let's first talk about some characteristics of RNA and some different kinds of RNA. And then probably next time we'll start off with, okay, how's transcription happen? Transcription and translation to me are a little bit more complicated than replication because you have to have a particular starting and stopping point as opposed to replication. All right, so structure of RNA, very similar to DNA, although it is single-stranded, of course, and DNA is always double-stranded. The exception being, and there's some viruses that have single-stranded DNA or double-stranded RNA, but at least we're talking about bacteria here. There are primary, secondary, and tertiary structures. So a primary structure, that just means the sequence of ribonucleotides, since we're talking about RNA. A secondary structure would be like a alpha helix or a beta sheet. And a tertiary structure is when you have some more complicated folding and bonding that takes place. So it depends on the kind of RNA that you're talking about. It has uracil as a base instead of thymine. Okay. And the other difference between RNA and DNA is that it has ribose sugar instead of deoxyribose. And so the only difference is that it has right here at the 2', the ribose sugar is an OH instead of just an H. So very similar in structure to DNA in a lot of ways. Okay, so what kinds of RNA are there? 3 different kinds of RNA. Of course, we already talked about RNA primers, necessary for DNA replication. Messenger RNA is the kind of RNA that kind of always first comes to my mind anyway, but this is only about 4% of the total RNA in the cell. It actually gets degraded pretty quickly. Okay. So some terminology that we use with messenger RNA. This term polycistronic in prokaryotes. And that just means that it encodes for multiple proteins. Okay, so we have one stretch of messenger RNA that when it's read at the ribosomes, we might get 3, 4, 5 different proteins from it. Okay, that's different than eukaryotes. Eukaryotes are monocistronic. That cistron means gene. Okay, so that just means encodes for one protein. Okay, so that's why in prokaryotes, we have operons. So we have genes that get transcribed together, genes that encode for proteins that maybe are all in the same pathway. So it's very an efficient way to kind of regulate the production of enzymes needed for a certain pathway. We don't really see that in eukaryotes. Of course, there's transfer RNA. That's about 10% of the cell's RNA. These are short pieces of RNA. They're about 75 nucleotides in length. And of course, their job is to deliver the correct amino acid that matches up with the messenger RNA to build proteins at the ribosome. All right, so if we just take a minute to look at the structure of a transfer RNA, we see it drawn as this kind of cloverleaf structure with these 3 hairpin loops. Up here, this is where the amino acid binds to the tRNA, and the really important region down here, we call this the anticodon, which matches up with codons on the messenger RNA. We'll see that later as we get into protein synthesis. Your book likes to draw tRNAs with this special kind of icon. Just that reminds us that when it's actually folded up the way it would be in a cell, it doesn't look, this is like if you put it flat out and then it's actually folded up in a way that looks more like this. So when you see that kind of bent icon, that's a tRNA. And the last kind of RNA is ribosomal RNA. And this is the most abundant type of RNA in the cell. It's the one that we tend to forget about, I think, or at least I do. It's the dominant molecule in the ribosome. Okay, so remember our ribosomal structure. There's segments of ribosomal RNA together with proteins. that make up the small and large subunit of the ribosome. One of the things to remember about ribosomal RNA is that it can be catalytic, meaning it can act as an enzyme. And when it does, we call it ribozyme. Okay. So that's kind of an interesting thing. It's ribozymes that initiate translation and initiate the bonding between the different amino acids. And interestingly enough, you know, that it's terribly complicated. So I just got this off of Wikipedia. This is the 16S strand of ribosomal RNA in a particular bacteria. Tremendously complicated. I think the ribosomal RNA is in tan and proteins are in purple down here. So just appreciate the complexity is all I'm saying. All right, so we will start out with transcription on Wednesday. Have a good day. I will be in my office most of the morning tomorrow if you want to come by and look at your exam.